Okay, maybe we should get started for the interest of time. So, good morning from Thailand, everyone. My name is Tiprat Chodibut, and it's a pleasure to welcome you to our first virtual research seminar during this global pandemic. I hope everyone is safe and healthy. This is a seminar jointly hosted by Chula Intelligent and Complex Systems Group and Chulalongkorn Cognitive Clinical and Computational Neuroscience Group, and also the Learning Institute from KMUTT. My name is Tiprat. I'm a faculty member of the physics department in Chulalongkorn University and a host for you today. I see many Thai colleagues in the area of computational neuroscience and physics joining, and also privileged to have a group of Professor Terry Sanowski from UCSD to join us today as well. And any of you who work on neural network definitely know of countless seminal contributions in neuroscience and machine learning by Professor Sanowski. So we are really excited and humbled to have you guys and your group to join us remotely today. So welcome everyone. And to those in the US, let me greet you in Thai way, Sawadee krap. So about the logistics, so this seminar will be recorded, and I'd like to defer the questions to the end of the talk. And although the seminar will be delivered in English because there will be some Thai colleagues joining, the questions are allowed to be asked in Thai. But if you need translation into English, you can ask us as well. And now I would like to pass the torch to Dr. Sirawat from KMUTT Learning Institute, who was a member of UCSD as well, to introduce our speaker, the first speaker of our virtual seminar series, and they are longtime friends and colleagues. So Dr. Sirawat himself also worked at UCSD, as you know. So please, Dr. Sirawat, I'll pass the stage to you now.
Hi everyone. So today is a great pleasure that I'm be able to introduce on one of the best student at UCSD. She just recently graduated. Now she's Dr. Rungratsameetaweemana. I have been working with her for a long time, 10 years plus now, and I've seen she grow so big. And I'm really excited that right now she's working with Claudia and Terry and doing some very exciting neuroscience stuff. And today I would give this stage to Nuttida Rungratsameetaweemana. Thank you.
Thank you, Ajahn Tip and Ajahn Sirawat. Thank you for the kind introduction. Again, before I begin my talk, I would like to mention that the work you will hear about today would not have been possible without the guidance and support from my amazing mentors who are joining us today, even though it is pretty late over here in the States. Dr. Terry Sanowski and Dr. Claudia Lanchek from the Computational Neurobiology Lab at the Salk Institute, and also Dr. Javi Garcia and Dr. Kanika Bansal from the Army Research Lab. Thank you. Okay, so today I will be talking about, so the title of my talk today is Uncovering Dynamical Chimera States in the Human Brain. Okay, let's see. Oh, sorry. Yeah, it was supposed to be that blind 2 minutes ago, but these are my mentors who are here with us today. They're amazing. And okay, anyway, so in Greek mythology, as a daughter of two serpents, Chimera is a fire-breathing hybrid creature, having a lion's head, a goat's body, and a serpent's tail. However, today the term chimera has come to refer to a surprising mathematical hybrid or a system with incongruent parts, such as a state of mixed synchronous and asynchronous behavior in a network of identical coupled oscillators. And the concept of chimeras has been applied to a number of systems across different fields. For example, recent work on cognitive chimeras by Dr. Bansal and Dr. Garcia shows that various cognitive functions can be explained in terms of different chimeras that are formed across brain regions. And by the end of my talk today, I hope to convince you that the concept of chimeras can also help us better understand the dynamics of epileptic seizures. So as an outline of my talk, we will first discuss background on epilepsy and the potential link between chimeras and this neurological disorder. And next, in part one, we'll discuss how a nonlinear dynamical system-based technique called delay differential analysis can be used to detect and predict epileptic seizures in humans. And in part two, we will then talk about how we can capture seizure and patient-specific information using network modeling. Okay, so let's start. Synchronization of neural activities, for example, in the hippocampus, is associated not only with not only with healthy brain functions, but also with neurological disorders such as epilepsy. And while neural synchronization, especially that associated with epilepsy, is often thought of as all or none, past work by Kuramoto and Strogatz suggests that the synchronization in the brain may in fact pass through an intermediate hybrid state where both synchrony and asynchrony coexist. And through the rest of my talk today, this state of partial synchrony or hybrid state will be referred to as a chimera state. Okay, so now let's talk about epilepsy. Epilepsy is a neurological disorder that is characterized by recurrent seizures and is caused by the excessive and disorganized spread of neural activities. Worldwide, more than 50 million people are living with epilepsy, and the seizures are defined as highly synchronized bursts of electrical activity in the brain, which cause involuntary alteration in behavior, movement, sensation, or consciousness. The unpredictable onset of seizures further burdens patients and their loved ones, as they can be prevented from performing everyday tasks such as driving. Therefore, precise prediction is key to developing a successful intervention for epilepsy. And while potential treatments do exist, medications could have severe side effects or, in fact, are shown to be ineffective in about 1/3 of patients. In addition, surgical resection could be risky and are not always effective. Therefore, developing effective strategies to predict and prevent epileptic seizures is one of the biggest challenges in basic and clinical neuroscience. So through mostly computational simulation and electronic circuit experiments, past studies have shown that chimera states can be observed in a variety of dynamical systems across a wide range of coupling topologies and coupling strings. And based on these findings, past work has proposed a possible connection between chimera states and epileptic seizures. However, to date, there's no direct evidence or neural evidence from epileptic patients that would support this account. And so the main goal of our project is to study chimera states and examine their links to epileptic seizures in humans. And just a side note here, substantial research has used various methods, including machine learning, advanced signal processing, and also other computational tools to try to predict seizures. However, to date, there is no clearly successful method. And so it is important that we understand the dynamics of neuronal activities that give rise to seizures. And one way to do that is through examining the possibility that the brain may pass through chimeras as it transitions from baseline normal state into a seizure state. So, to study the intricate dynamics of chimeras in the brain, we use a novel analytical technique developed by Dr. Lanchek, called delay differential analysis, or DDA. So DDA is a nonlinear method that combines two types of embeddings, the delay embedding and differential embedding. So for example, here, in the bottom, we have the unknown system that we would like to study. For example, this is, for example, human brain. And so X here represents a time series signal recorded from our unknown system, or the brain in this case. So on the left here, we have delay embedding, where we essentially plot the original time series and its delay version of itself. And on the right here, we have differential embedding, where we plot the original time series X over its numerically differential version. And you can see that the structures are pretty similar. And what DDA tries to do is to find a relationship between these two types of embeddings. So conceptually, you can think of DDA as a way to model, is a way of modeling changes in the system dynamics as represented by the differential embedding. So that's, so the differential embedding represents the dynamical changes in the system. So the DDA is a way of modeling that change in terms of what happened earlier in time, and that is represented by the delay embedding term. And so here we have the mathematical formulation of DDA. Differential embedding is shown on the left here, and delay embedding is on the right. We use a three-term model as shown here. Again, on the left side, we have the numerically differential version of X, and on the right side, we have a couple terms of the delay versions of the original time series. So delayed by tau 1 and tau 2. And we also have a nonlinear term, x squared, as well. So the DDA finds the model coefficients A1, A2, and A3, along with that best relate the delay embeddings to the differential embedding. And these coefficients A1, A2, A3, along with the least square error row, are called DDA features. And these features can be used to identify dynamical differences in data. And now we can apply our DDA model to neuro time series data as recorded from patients with epilepsy through intracranial recordings. And before we dive into the results, I would like to take a step back and give an overview of DDA pipeline in comparison to other more traditional signal processing methods. So for traditional analysis, data are usually pre-processed, and spectral features can be computed. So these features then go through dimensionality reduction, which give out informative features that can be used to, for example, differentiate data from experimental condition A from those of experimental condition B. And these steps are repeated each time as a new set of data is obtained. On the other hand, here we have the delay differential analysis. So for the DDA, there are two parts to its pipeline: the structure selection and data analysis. So the assumption of the structure selection step here is that the global dynamics that underlie a biological system can consistently be best captured or best explained by one DDA model. And so in other words, what that means is that during this step, we can run through all the possible variations of DDA models. So with different numbers of linear and nonlinear terms, and also with different values of time delay or taus. And we can then find the model that can best explain the differences, say, between seizure and non-seizure data from patients with epilepsy. So in the case of epilepsy, this three-term model turned out to be the most parsimonious. And the best fitting delay pairs are 7 and 10, which essentially means that for, say, epilepsy data with a resampling rate of 500 Hertz, it's most informative to look at the past values of the system at 14 and 20 milliseconds. So keep in mind that the structure selection step, the structure selection step here only needs to be done once per data type. And so as long as we are analyzing epilepsy data, we will not have to redo this step. However, if, for example, we want to study cardiography data, we will have to perform structural selection to find the best model, which could be different from the 3-term model shown here for that data type. And so in the next step, data analysis, we simply take the best model for our data type and compute a model feature A1, A2, and A3 and row in this case. And past studies have shown that these DDA features carry information about the underlying dynamics of various biological systems. And in this part of the talk, we will be examining dynamics of epileptic seizures through the DDA feature A1, so the very first coefficient here. Okay, so here we are looking at, so again here in the left corner we have the equation that or the model that best explains epilepsy data in general. So essentially we fit this model to our intracranial recording data. And here we are looking at DDA feature A1 corresponding to each of the 77 recording channels over time. So on the y-axis we have the recording channel numbers. The top panel shows A1 over 100 hours of recording, and the bottom panel zooms in on the hour 100 to 120 of recording, during which all seven seizures within this data set of this patient occurred. And the recording channels are sorted on the y-axis here, such that the seizure onset channels, as determined by a neurologist, are shown here as channel 1 to 32, and the non-onset channels are shown here as channel 33 to 77. So the onset of each of the seven seizures are indicated by the pink arrows, both the top plot and the bottom plot. So we can see that there are some patterns in our A1 parameter that consistently emerge prior to the onset of each seizures. Right, and interestingly, these patterns can only be seen within the seizure onset channels. So next, we further examine these patterns by looking at the phases of A1 over time. Again, we're looking at A1. So we're looking at the phases of A1 over time, and also as a function of recording channels, the sorted recording channels. Again, we see these patterns within the seizure onset channels that reliably appear before each seizure onset, but not anywhere else in time. Next, let us take a closer look at these patterns by taking 2 time points. So we're gonna take a look at just one seizure, seizure number 4, and at 2 time points. 10 minutes before and 10 minutes after the 4th seizure. So here we are plotting the phases of A1 at those 2 time points. Again, 10 minutes before the onset of this seizure and 10 minutes after. Just like before, the recording channels are sorted such that the first 32 channels are seizure onset channels. At 10 minutes before the seizure onset, desynchronization of the onset channels can be seen with coexisting synchronization within the non-onset channels. After 10 minutes, so at 10 minutes after the seizure, the chimera state, as we saw before, is gone. And we instead observe synchronization across the whole brain, or essentially across all recording channels. So now, so this is one seizure, one sample of seizures. So now, let me play this video. So now let's look at how the brain transitions in and out of these chimera states as a seizure is developing in real time. So we are looking at the fourth seizure again from 5 minutes before to 5 minutes after the onset. And the phases of A1 over time is shown in the bottom plot. And these phases values correspond to each recording channels at each time point. The phases values corresponding to each time point are being plotted in the top panel. A dotted pink line indicates the onset of the seizure marked by the neurologist. And we can see that leading up to the onset, chimera states can be observed, where the seizure onset channels desynchronize and the non-onset channels stay synchronized. And now during the seizures, these patterns of behavior change, and we in fact do see a variety of chaotic behavior. And right here, now after the seizure terminates, we observe a complete synchronization across all channels. Okay, so let me move to the next slide. Okay, so to systematically identify chimera states, we need to be able to quantify the level of synchrony observed in our data, or specifically in the phases of A1. And one way to do that is to compute what's called Kuramoto order parameter. And as an example, let's take a zoom-in version of the fourth seizure one more time, where the dotted line indicates the onset of this seizure. The bottom plot shows mean Kuramoto order parameter, where 1 indicates complete order or complete synchrony or synchronization, and 0 indicates no synchronization. The lowest order parameter value is observed during the seizure, followed by the time segments before the seizure during which chimera states are observed. So actually, our results do suggest that the true onset of this seizure, for example, may in fact happen a few seconds after what has been indicated by the neurologist. Note that this pattern of activity in the phases of A1, as seen here in the top plot, was also observed in all of the 7 seizures in this patient. Here on the left, so each panel shows the phases of A1 corresponding to each of the 7 seizures of this patient. And so as seen here on the left, A1 phases of each of the 7 seizures are plotted from 30 minutes before to 30 minutes after the onset. And on the right, we have just zoom-in versions of 5 minutes before to 5 minutes after the onset. So phases of all 7 seizures are centered around these seizure onsets as indicated by the neurologist. And we can strikingly similar patterns can be observed across all of these 7 seizures. Right, so finally, we also introduce an alternative way to Kuramoto order parameter that can quantify the level of synchrony in our system by directly computing the synchrony measure directly from our DDA feature A1. And that is called DDA order parameter. So we computed DDA order parameter, which is essentially a mean of pairwise L2 norm or Euclidean distances between A1 corresponding to each recording channel. And so we did so in a 5-second sliding windows with a shift of 250 milliseconds. And a smaller pairwise difference indicates higher degree of similarity or synchronization between channels. And we can see that our DDA order parameters closely track the dynamics of chimera states leading up to each of the 7 seizure onset. Finally, we can also look at the DDA order parameter as a function of recording channels. So here we are plotting over time the channels whose mean pairwise Euclidean distances to other channels are in the lower 30%. we then normalize this mean Euclidean distance, or the DDA parameter, such that in a chimera state, so we can take a look at the zoom-in plot in the bottom. So in a chimera state, the synchronized channels have values that are between 0.5 to 1, as indicated by the dark to light brown here. Okay. And during this chimera state, the desynchronized channels will have values between 0 to 0.5, as indicated by light to dark gray here. So in a non-chimera state, where all recording channels display either synchronous or asynchronous behavior, the normalized DDA order parameter will be of values 1 or 0, and they are indicated by the light color on this in this plot. And our DDA order parameter measure further confirms the consistent patterns of the coexistence of desynchronized behavior in the onset channels, along with the synchronized behavior in the non-onset channel prior to each of the 7 seizure onset. So far, we have shown that in one patient, each seizure was reliably preceded by chimera states that lasted up to 2 hours, suggesting a new feature that can potentially be used for long-term seizure prediction in some patients. And this is, to the best of our knowledge, the first direct evidence of chimera states in the human epileptic brain. Next, so again, here I'm showing the DDA model that we used previously. So next, we then apply this exact 3-term DDA model with a fixed delay pair of 7 and 10, as mentioned before, to data recorded from now other patients. And even though we were able to detect some chimera states leading up to a seizure, these detected states occur much closer in time to the actual seizure onset than what we previously saw in our first patient. So can we do better? It is possible that there are some patient or even seizure specific information that could lead to different temporal dynamics of seizures across patients. And so instead of using just a fixed delay pair, we can now vary the values of tau 1 and tau 2 to be from, say, 1 to 60. And what that means is that we're asking if we could capture extended chimera states in other patients by looking at past dynamics of the system at different time points or different points in time. And if so, what are the most informative time windows for predicting seizures in each patient specifically? And finally, we can then ask if we can use these informative time windows as additional method for seizure prediction, for seizure subtype classification in the future. Here I'm showing a schematic of what we did. So here, to remind you, we have our DDA model with tau 1 and tau 2 as our delay parameters. So in the previous part, we fixed this tau 1 and tau 2 to be at 7 and 10, and because that was the best fitting taus across patients as we found from the structure selection step, right? So essentially, we used tau 1 and tau 2. With this 3-term model, we apply the model to the intracranial recording from each patient, and we get out something like this. So say we have these phases of A1 over time, and what we can do is we can compute the mean DDA order parameter, which captures the level of synchronization corresponding to each recording channel within each time window of interest. And then we can also define what time period we are interested in looking at in relation to the seizure onset. For example, we can say we want to look we want to look far away earlier than when the seizure happens, and then in the second time window we want to look closer to the seizure onset, and then we can also look at the time window after, way after the seizure terminates. And then we can plot that mean, so we will then get one mean DDA order parameter value as shown in as a yellow dot here, as per one pair of tau 1 and tau 2, right? So in our previous analysis, we use only one pair of tau 1 and tau 2. So essentially for each of this time window, we only get one value of the mean DDA order parameter, right? That is a mean over time and channel within that time window. So now in this part of the analysis, we want to explore, or we want to sample the delay, the time delay space. So instead of using a fixed value for tau 1 and tau 2, we varied the value to be from 1 to 60. So then what I will show in the next plot would be a lot of dots here, right? So one dot corresponding to each delay pair. So tau 1 will go from 1 to 60, and tau 2 will also go to from 1 to 60, and you will see a lot of dots in the next slide. Okay, and then also we're looking at 3 time windows. So one from way before the seizure onset. The second one would be close to close to seizure onset, and the third one would be after the seizure onset. Okay, and I will be showing the sample plot of one patient for simplicity. If I can move to the next slide. Okay, so here we're showing again the 3 time windows. The left one shows the early time window, 48 minutes to 30 minutes before the onset, and the middle one shows 20 minutes before the onset. And the third panel to the right here shows the results from 9 minutes after the seizure. So, and here essentially I'm plotting the mean of the DDA order parameter as shown as a yellow dot in the previous slide, right, corresponding to each combination of tau 1 and tau 2. And on the bottom plot here, I'm essentially just showing only the top performing taus at capturing the chimera state. So let me, so at capturing the chimera states here. So we can see that the chimera states are can be captured or can be seen only during this time window, like 20 minutes before the seizure onset. But you can see that this emergence of chimera states can be seen by using a variety of different combinations of tau 1 and tau 2. Next, we can compare the performance of DDA order parameter at detecting chimera states with the Kuramoto order parameter, right? And what the takeaway point from this slide is that we can reliably see the chimera states that are picked up by this analysis only prior to the seizure onset, but not during the other 2 time windows. But when we use the Kuramoto order parameter, we found that the results are less clear, and it's a little bit hard to tell where the chimera state can be detected. So together, this shows a potential use of DDA order parameter in detection of chimera states that lead up to epileptic seizures, even in patients that we could not really detect the extended chimera state using a fixed delay pair in the previous part. And so, right, so now we can further try to understand or capture the seizure or patient-specific information using a different method borrowed from network science. And to to simulate an epileptic network, we adapted a computational model which has previously been proposed to capture certain aspects of epileptic data in humans. And the model dynamics the model dynamics are based on the hypothesis that the change in the brain states the change in the brain state that causes seizure is driven by noise in a bistable system. And so in a network, the individual node dynamics are governed by the stochastic complex differential equation as shown here, where lambda determines the possible attractors of the system, and the stochastic noise, eta, represents the complex noise input to the model, which incorporated the white noise to imitate state transitions driven by both external and endogenous factors. And so individual nodes in a network of n nodes are connected with one another through through bidirectional functional connectivity C here to form a network. And when we consider the deterministic dynamics of the model, that is without the noise or the connectivity term, we see that different initial conditions can push this network into either a resting or a fixed point state or an oscillating state. And so plotted here is a bifurcation in the lambda parameter. And two states are separated by an unstable oscillation. And the model parameters were chosen such that all nodes in the model are placed in the bistable regime. Okay. And by introducing eta or the complex noise term, the nodes exhibited occasional transitions between the resting state and the oscillating state. Right. Individually, each node in this bistable setting of the model parameters produces a simulated time series with a resting state and episodes of pathological high amplitude of oscillations. And these oscillations are identified with seizure dynamics, whereas the resting state is identifying with non-seizure dynamics. And so now to construct a patient-specific network, we infer network, so see here, we infer a network connectivity matrix from a non-seizure period of the recordings or of the data of each patient, and use this connectivity matrix as the network structure in our model, specific for each patient. And within this framework, within this modeling framework, we consider the cortical region under each recording channel or recording electrode as a network node. And so if we have data recorded from 77 intracranial electrodes, then we can build a model with 77 nodes to represent the 77 cortical region. And so now our C here will represent a patient-specific connectivity, which reflects the functional interactions between different cortical regions. And we can then, so we can compute this connectivity matrix from the non-seizure period in the intracranial data, which will reflect the patient's baseline functional connectivity. And when we would like to examine seizure dynamics, we can then instead compute this connectivity matrix based on the seizure period in the data and study the network model behavior. So, so here the dynamics leading to a seizure onset can be quantified using the time taken by a node to leave the basin of attraction in the resting state and cross over to the basin of attraction of the oscillating state. oscillating seizure state. And here on the left, we have a control dynamics computed as a mean network models based on non-seizure data from 10 patients. And on the right, we have 3 examples of seizure dynamics from 3 different patients. We can see that overall, it is more likely for a node within, so on the y-axis, we have node here, we're plotting the recording channels or node here. And on the x-axis is over time of the model. So we show that overall, it is more likely for a node within the seizure networks to go into oscillating or seizure state as compared to that within the control network. And the likelihood of a node to go into seizure state is inversely related to this transition time, which which essentially tells us about how stable or unstable the network is. And so to quantify this, we can use, for example, some graph theory measures. Here we're using clustering coefficients and synchronizability measures to essentially quantify the differences we observed in the likelihood of a node to go into seizure state, or the transition time it takes for the network to oscillate. So we show that the mean transition time within the control network is longer than that of each of the seizure network of each patient, each seizure of each patient. And our next step will be to look at how the transition time of each recording channel in this seizure networks contributes to this effect. And this could help us better understand the spatial dynamics that are specific to each seizure and each patient, which can potentially be used to complement our DDA features for seizure prediction as well as seizure subtype classification in the future. And so, right, I hope, right, so yeah, by simulating epileptic models, we show that seizure activities can be detected through the level of instability or the transition time within the network, the simulated network. And so I hope I have convinced you today that we can use the concept, we can apply the concept of chimeras to help us understand the neurodynamics that underlie epileptic seizures. So we found chimera states in 1 patient that reliably preceded each of the 7 seizures up to 2 hours prior to the onset. So this is from part 1. And then we also developed a DDA order parameter, which can automatically detect chimera states and capture seizure and patient-specific information that could be used for potential classification of seizure subtypes. And finally, by simulating a network model that incorporates patient-specific functional connectivity, we also show that seizure dynamics can be further indexed through the level of instability in each network. And so, yep, I hope you are all convinced that we have added this one more thing we could study with the concept of chimeras to our list of chimeras being spotted in the real world. And right, so I would like to thank Ajahn Tip and Ajahn Sirawash and everybody for the kind invitation to include me as a part of this wonderful seminar series. I would like to also thank Dr. Sydney Cash from Harvard Medical School and Massachusetts General Hospital for providing intracranial data and for all the helpful discussion. I would also like to thank my mentors, Terry, Claudia, Javi, and Kanika, my colleagues and my friends, a lot of whom made time to join us today. And finally, thank you everybody for your interest in our work. I will now take questions. Thank you.
All right, I'll give a virtual clap. Thank you very much.
Thank you. That's the real clap. Thank you.
Wonderful talk. I think now we can open the floor for questions. So you can unmute yourself or raise hands. I'll try to unmute you and open the floor for discussions. Okay, maybe I shall start. Some people are still shy over here. So, so in the DDA model, there seems to be 3 parameters, right? A1, A2, and A3.
Yep.
And in particular, you look at only the quadratic terms that depends on X1. Is there any good reason why you neglect X2 in there?
So so what we show today is based on A1. Yeah, that's right. So A1 is right here. So it's the linear term. It's the coefficient of the first linear term. And so we found that we also see similar patterns, but less so through A2 and A3. And that's why, so A1 seems to capture the most information, and that's why we decided to show A1. But it's also possible that in some other data type, you might also see information using other coefficients. But yeah, essentially we use A1 because it captures the most information in this case.
Okay, and how does different nodes couple to each other? This seems to be just one variable, right?
For X. So for this one, if we can look, so if we, okay, maybe I can show this graph. Yeah, so for each channel, so we got, actually, we go back to the schematic. So here, from each recording channel, right, we get 1 time series data. We get 1 time series signal, and then we can fit individual channel using the model. So you get like A1 for channel 1, A2 for channel 2.
Okay.
A for channel 3, yeah.
Okay, I see.
Yeah.
But are there
like
coupling between different channels, or from the equation it seems that it's independent of each other?
Yeah. So yeah, that's a good question. So I mean, you mean coupling by like within the result of the A1, or?
Yeah, like X at one location coupled to the nearby locations.
Yeah, so in this part, we essentially just fit the model individually for each individual channel.
Okay.
But there is an extension of this model called cross dynamic delay differential analysis, or CDDDA, that Chris and Claudia in the lab have been working on, which can give you more information about the direction of information flow across channels, for example, in this case. So by using that, you can capture more coupling information, for example, or spatial information, or direction of information flow across channels, which will also be useful for seizure prediction as well. Yep.
Okay, I see. So the parameter A1 sort of different from channel to channel, I guess?
Yeah, so you get 1 A1 from each, so you get
channel.
1 A1 for each time series, depending on where. Yeah, so if you do one time, if you treat the one time series signal from one channel, then you fit it once, right? And you get 1 value, A1.
Okay.
Yep.
Thank you.
Thank you.
If anyone else has question, please unmute yourself and then ask.
Hello. So I actually have a question. Hi.
Yeah.
So I'm Pantita. I'm actually a quantum physicist, so I'm a complete ignoramus in what you do. My question is probably very basic here. So you mentioned you use DDA as your model. I'm just wondering if there's any other model that other people use in the field to kind of model the signal for the brain.
Yeah, so right, so there is substantial research, right, on trying to predict seizures, and a lot of signal processing methods have been used. And for example, some people have tried to model the dynamics of seizures using spectral features, for example, gamma activity, or within specific area, like maybe like frontal or parietal, depending on, I guess, the type of seizures you're trying to model. But essentially, you could use other features, right? You can maybe use spectral features of interest to try to model the seizures, and people have done that, and people have also tried using machine learning as well to predict seizures. But right, so the reason why we use DDA in our case is because it is computationally inexpensive, right? Because we can skip that whole dimensionality reduction and we can just go right to the data analysis part. So we can pretty quickly go through a lot of data over over short time period, and we essentially compress data, right? We compress the data as opposed to like blowing up, expanding the data into various features. So that's one part of it. And the second part is that the DDA captures nonlinear information, right? Which other linear methods could be applied to. And since chimera states have not been seen before in epilepsy, and so we are not sure, but if we were to just use linear method, we might miss some information, right? So it's possible that to be able to capture like chimera states, we might need also like to look at nonlinear dynamics as well. And that's why we went with DDA analysis.
I see.
I hope that answered your question.
Yes, just a little bit of follow-up. So I'm gathering a little bit from your answer that it might be possible to see chimera state from other model.
Yeah, so we have not ourselves tried using other methods to see if we could see chimera states or not. Yeah.
But for sure that it's possible to see from DDA.
Yeah. So based on based on what we found, we think that at least in one patient, right, using just fixed value of delay pair, we could consistently see that. And now we're trying to see if we could also detect chimera states consistently across patients by varying like how far in the past we want to look at the past values of the system. Yep.
Okay. Thank you.
So I have two questions. So one is a very naive one. I wonder, like, if you do dimensionality reduction here using just a linear equation, what would happen?
I'm not sure I understand the question. Sorry.
So if you use just a linear equation here and not a linear equation, what would happen?
Okay. So essentially dimensionality reduction. So in this part, right, the feature selection. So what we did is the goal of structure selection is to try to find the best model that will explain your data type. Okay? So you can imagine that it could be like x to the third for one data type, maybe, right? That would best explain. And so this process is essentially running through every single possible model just to find the best model that give you the best classification accuracy.
I see.
So I guess it's not.
Model.
So yes, you could also, yeah. So you're running through, if that's one of the possible models that you're interested in, right? You want to see if that will explain the data type that you have, then you would include that in your process.
I see. So if you go back,
but if you only have just two terms, right? If you only have just the linears, that would be similar to like spectral analysis, right? That we are used to doing. Yeah.
Okay. Can you go back like toward the end? I'm sorry.
Go back. Forward. Go back. Forward.
Forward.
Yes. Can you tell me which part?
Like a bifurcating.
Okay.
More.
This is.
Yeah, and more. Yeah. Oh, I get one more.
Yeah. One more.
Okay. There is. Yeah. So I kind of wonder why, in control, there's that oscillation in the back.
Okay. So first, this is preliminary work and a lot still has to be done, right? But okay, so you're asking why does this oscillating state, right? When this is a control network. Yeah, because that is how the model is set up. So the model is set up such that, like, you know, by adding some noise, the complex noise, you and you know, given that we chose the model parameters such that the model would be in the bistable regime, by adding some noise, at some point you are gonna push the network into the oscillating state just by how the model is set up. So you can have the network going into oscillating state, even though you're, even with the C term here, the functional connectivity that is taken from the non-seizure period, just because this is how the model is set up, right? But the informative feature, or the take-home point is that what you would like to see, you would like to compare, what you want to see is you would like to see the difference in the transition time, essentially, of the network to go from resting state into oscillating state, right? To go, like we have bistable switch, right? To go from 1 to 2, like how long does it take, how stable or how resilient the model is, right? In that transition, right? And we just essentially show that when using different network structure, whether taken, you know, from the non-seizure data or the seizure data, we can capture the difference in the, like how stable the network is or the model is.
So, okay, good, thank you. So I have a last question, sorry. So the last question is, are you doing this with EEG, and what do you expect with EEG? And the next, the following question is, so what is the next science question that would come up?
Okay, so the first question with EEG as in, like,
not ECOG, right? Just like scalp EEG. Is it scalp EEG or ECOG?
Intracranial.
This ECOG, right? So it
so,
would be with EEG?
Yeah, so I mean, because you mean this modeling part, right? So because we derive, so for each patient, right, to construct a patient-specific model, right, we derive, or we extracted the connectivity matrix from the, like the spatial, essentially the spatial information, right, within the data. So now if you're, instead of using intracranial, you're using EEG, you're now having less spatial information, right? Or less informative connectivity matrix, I would think. So then when you have connectivity, so during seizure period, when you have network connectivity matrix that is not super precise, then the difference when you are comparing your network, your control network to your seizure network, then you should not see as big of a difference, because your functional connectivity through EEG is not as precise, or it doesn't contain as much information, like coupling information.
The how big is the location of the seizures, like how expand, like the,
yeah, so
is this or something like that.
Yeah, so that is a part of my answer to your second question. So we can then use, so you can see that there's some interesting results here, right, associated with different nodes, right? So what we can do next is we can then look at the contribution, or how each of the location of each of these nodes on the patient's brain, and how they contribute to the overall, like, mean transition time, right? So what is the difference between, say, that node from that node? Maybe one, like, you know, we haven't really sorted the channels based on, for this analysis, based on the location, like how far each electrode is, for example, from the epilepsy zone, as indicated by neurologists, right, for example. So there is possible information regarding, like, the spatial contribution of different channels that could be extracted or further examined using this network modeling, and that could help also with the subtype classification of seizures, because you can have different origins of seizures from different brain regions, right? That we could potentially study by looking at individual nodes' contribution to the transition time.
Thank you.
Thank you.
Okay, any more questions from the audience?
Hi, I have a quick question. Maybe you already mentioned this, but how did you determine the phase of A1? Is it a complex parameter or no, it's time dependent, right?
Hilbert transform.
Oh, okay.
Yeah.
All right, thank you.
Yeah.
I have a question actually. If I
Yes.
could follow up on Sirwaj's question on porting this to EEG, like scalp EEG. So your answer seemed to imply that basically it would be a little noisier given like a functional connectivity network derived from the EEG. So what if you essentially like mixed methods? Like say getting you know, a very stable kind of even structural network from, you know, DSI or something, and then transferring your EEG activity, you know, using a source analysis or something to particular sources on the scalp, using a boundary element model or something. Do you think that simply would be a noisier measurement and that you get like similar kind of predictivity? Or what do you think? Just what's your hunch?
Yeah, that would definitely be better than just scalp EEG, I think. But I mean, I do think that the intracranial recording would still give the best, like the most like precise. It's closest to the ground truth of the actual network, an actual epileptic network. But yeah, I have not thought about using like, you know, combining like scalp EEG with other methods, as you mentioned. That could potentially boost a lot of like information as compared to like just EEG, for sure. The other thing is, so we don't have the same like number of channels for EEG and like intracranial recordings channels within patient. So it could be a little hard to directly compare, you know, like because we would have,
Nuttida?
Yeah.
Yeah, so at MGH in Boston, I looked at, for Alzheimer's patients, I looked at seizures from scalp EEG and from ECOG. And you see the seizures also from EEG, but not as strong from ECOG. So I tried it, but since we have so much data, ECOG data, I think we first should just concentrate on the ECOG data. But EEG, you can do it, but not as well as ECOG.
Right. Yeah, and right. So yeah, so I guess to answer Javi's question, I think a part of it is because of this like spatial information, right? It's like further from the origin of the seizure, so more noise. But yeah, like to use other methods like DSI or other maybe potentially other computational methods to try to correct for that could help. I'm not sure, like, you know, to what the extent would be. But essentially, we could try to use other methods to correct for that distance, you know, in space.
Or perhaps the target of prediction may be just a little looser target, right? Like seizure is such like an abrupt event, right? And it has like dramatic sort of patient consequences, right? So if you're using this in another sort of, you know, domain, like maybe you can be a little more flexible about your prediction, essentially.
Yeah.
I kind of wonder, do you know this disease called RVD in PD patient?
No.
So it's like a REM sleep disorder where people just like have like this erratic movement when they're sleeping in some of Parkinson's patients. Yeah, so
I see.
yeah, I kind of like, I think we like CHIMERA has data. We have the data at Chulalongkorn University, having recording like people sleeping and then have this RBD movement thing. I'm not really quite sure, but it's all EEG and EMG and stuff like that. I'm kind of wondering like maybe this method could be really helpful, and also at CHIMERAS Hospital, I would say CHIMERAS Hospital, you have a lot of EEG data on, like in epileptic patients, right? So I can see this as like a very good resource for a ton of data that we can use in Thailand. I see a lot of potentials here, right, Shaipat and Tiprat?
Thank you.
I suppose I should say something. Hi, Nuttida. I missed your talk. I'm so sorry. Today was a bit hectic for me. But I guess I read some of your paper about this topic, and I wonder what type of what is the etiology of this epilepsy patient? You said it's only in this particular patients, right, that you find this phenomena.
Yeah. So that's a question that we would also like to examine in the future, right? And we would need, so essentially we're asking, can we basically cluster or classify, right, patients into based on their etiology of the disease. It's a little late here, sorry. Based on like the pattern of results that we can see, right? So maybe if we have other patients that have similar like origins of seizures, maybe we will see more similar patterns of like CHIMERA states, for example, right? So like we could use after trying this method on more patients, then we can try to cluster patients based on the results, the similarity, right, across results.
Right, that would be interesting. But one of the reasons could be that we don't have a whole brain coverage, right? So it could be that you find this specifically in these patients only because we're lucky enough to cover enough to see this in this patient. So I wonder if you have extra coverage in this patient or just by luck or something.
Yeah, that's a good question. So for the patient that so in the second part of in the network model part, we show like results from a patient with even more coverage actually.
Gotcha.
So this patient, we had 80 channel, so a couple more than in the previous one, right? But for this patient, we using just like fixed value of like tau 1 and tau 2, we could not really capture like extended CHIMERA states, right? And so, but when we try using other values of tau 1 and tau 2, we seem to be able to capture CHIMERA states. So it's possible, like having a lot of coverage is always better, but also when we don't see it, right, it's possible that we're not looking at the right time, right? There are it's possible that there are like the most informative time windows at which we should look at, right, the past value of the system that we should look at, that could be specific for each patient depending on the type of seizures they have as well. So that's another kind of layer that or feature that we could use for to classify some types of seizure as well.
Right.
So basically more patients that I need to,
It's super impressive that you can predict, I think, 2 hours before the seizure.
Yeah, 2 hours.
If I remember correctly, like hours before the seizure. Patient, so it's a lot of time window that you can, tell patients to up the medication or something to prevent
Right.
seizure.
Right.
But to go forward, do you think it's possible that we can, like, my question would be, is it possible to reduce the number of contacts that we need to put electrodes on the brain? Like, what is the minimum number? Is it possible that we can, recommend patients to, let's say, we implant couples of electrodes to capture this event? Is it possible, or you need a bunch of electrodes?
So I think for the initial state of trying to understand, right, like when or in which type of data or which type of patients we can see extended chimera states. Like this first part, we're still trying to understand, right, like how to best detect this. We want the chimera states to, we want to see them, and we want them to be as long as possible, right? We want to be able to predict the longest possible, like transitional
You don't want to
states
throw anything away
to maximize
at the beginning,
right? To maximize possibility of intervention. But once we have better understanding of what
then
factors
you can reduce,
contribute, then potentially then we may just need the informative channels, whatever they may be, for the subtype.
So that would definitely be interesting in terms of the practical point, critical part of this work.
This is surprising.
Sometimes 3 or 4 channels are completely enough.
Yeah, I think I'm missing something regarding tau 1 and tau 2. Like, for example, on the slide you're plotting in the middle lower one, it seems like there's specific value of tau 1 and tau 2 where you can observe chimera states. Is that right? And the question is, in the modeling part, when you get the data, does it mean that tau 1 and tau 2 changes over time, or what does it mean for one channel?
For one channel. So 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 let me just go back just to the structure selection part. So so for this structure selection part, right, before we get that model, right, we try a bunch of different models, right, for our data type, and we're asking which what variant of the DDA model would best explain our data type, which is epilepsy data in this case, right?
So
we
from
found
what I
that
understand, it seems like
right?
one channel has one variable X associated to it, but there are 2 time delays in one channel, and you try to effectively capture the coupling through this A1, A2, and A3. Was that what I'm understanding?
Right. So so if we look here, right, let's say that this is the time, X is the time series or the data collected from one channel, right? And so over time, this is data or signal recorded from channel A over time. Let's just say channel M over time. And so when we fit the model, the 3-term model, to this X signal, right, we get A1, A2, and A3. Right, and that value, you get one value, right? It doesn't change over time.
Right, so you have to choose tau 1, tau 2, and for each tau 1, tau 2, you get the best fit A1, A2, A3.
Yes, so right. So tau 1 and tau 2 are a part of the best model, basically. So this is the best model, right? And in this case, tau 1 was 7 and tau 2 was 10, right? And so that comes as a part of the best model for epilepsy data. But in the second part, we're like foregoing this information. We're not using this information because we're thinking that, maybe there's some patient-specific dynamics that heavily drive these chimera states. And so by fixing the time in the past where we want to look at the data, right? We like the time we would like to shift the original time series, right? By fixing that value to be just 7 and 10 and using that across all patients without considering individual differences, we might miss, right, the chimera states.
Okay, and the only nonlinearity comes from coupling to the past by itself, right? There's no different channels coupling at all, like X1.
So not for this one. We get each we fit yeah, we fit the model with the time series taken from each channel for this one, right? We get A1, A2, A3, and row for each time series, which is taken from each channel.
I see.
Yeah.
I see. Okay. And from the DDA model, I'm curious what happens to x2 square. As a physicist, when you see some symmetry broken, you just start to wonder why do you neglect tau 2 in there. Where's x2 square?
Yeah, so like in this part, right? So like this is the like most parsimonious model, right? Which means that if you can add more terms, you can add probably like in some data type, right? You can imagine where you add x tau 2 square, and you can capture just as much information, which means that we don't need that, right? For if you for this state for like epilepsy, by adding more terms, we don't capture any more information essentially, and that's why we just stick with the 3 term. But there could be some data type where you would need more quadratic terms, or maybe
I would guess if
sure.
there's a clear separation of time scale between T1 and T2, then the coupling with xt2 square might not be so important. But if T1 and T2 is off the same scale, like the same millisecond, then it might be. But it seems from your model that this is fine enough. This is good enough.
Okay. Thank you.
Other questions from the audience?
I have a beginner question.
Yes.
I like to know that, okay, from what I can understand is that the CHIMERA state precedes seizures, right?
Yes.
And do we know the causation between the CHIMERA state and seizures? Or they are just correlated?
You mean, is it possible to have chimera states and not seizures? Is that one way of
Not really. I like to understand more about CHIMERA state, why it causes seizures, if it does.
See. Yeah. So I think one way that I like to think about this is that when you imagine like you have, so without seeing any data, right? So when you think of a brain, right, at rest, a system in a resting state, you have, all the different nodes doing different things, right? And when seizures happen, you have like this really highly synchronous behavior across all channels. So to go from highly disorganized or highly desynchronized behavior to highly synchronized, it's logical, right, to think that the brain would have to go through this like intermediate state. So I would not really call like this intermediate state as as it causing the third state, but I would think of it as a more like transition, a logical transition point, right, to go from highly desynchronized to highly synchronized behavior. So it's more like an intermediate state that the brain goes through, and the fact that we could detect that helps us predict what's coming. So I would not really think of it in terms of like causality per se, but more of like a different state, like yeah.
Okay. Can I ask that? Do we know the cause of seizures? That what causes the change between the conformity and non-conformity states?
Yeah, is tripod still here? Okay, I guess it's not here. So you mean right? So what I know is that, right, you can have like one cortical region that has abnormal neuronal function or neuronal activities, right? And because that cortical region connects, right, connects to other cortical regions, that information or that excessive or abnormal neuronal activation is now spread to other channels. And so the starts from like the neighboring cortical regions would have this abnormal activities, and that can spread to the rest of the brain, and that cause like seizures.
Okay, yep, that's pretty precise. So yeah, I'm listening. Yep, so it depends on the type of the epilepsy. We have focal type and generalized. For the focal type, normally it starts at very tiny patch of cortex, and then it spread to other regions. It doesn't have to be the adjacent region, because it could be connected via the white matter tract, and then sometimes you see it spread from one side to the other without going to the neighbor on the same side. That could happen too. So it depends on the location of the epileptogenic zone, we call.
Mm-hmm.
Is that the question? I assume so.
Yes,
okay.
It is the question.
Thank you.
Okay. Thank you so much.
So maybe I can try to understand the first part of your talk this way. Please correct me if I'm wrong. I was trying to understand how you approach this epileptic seizure by doing dimensionality reduction of the models, where different patch or your different EEG channel can be described by this delay differential analysis independently of other channels, right? And then when the seizures happen, you do some sort of maximum likelihood estimate of the parameters and see that the dynamics split into two groups, the groups with the same phase, right? Two different phases.
Mm-hmm.
So the question is, does those parameters change over time, or A1, A2, for example? So in principle, it should change, right? Because the seizure state and pre-seizure state, they should have different dynamics. Or maybe I understand it completely wrong.
Right. So when we fit the model, we did the, I see, okay. I understand what you're asking now. So we did like the sliding windows, right? So we have like, so we have like hours of recording, say like 120 hours of recording, right? So we fit the model with like sliding window, like small chunks of data. So it does change over time depending on what state, I guess, the brain was in, yeah.
Okay, I see.
But essentially during like chimera states, right, that what we call chimera states, we see this like split of behavior or coexisting of different behaviors in the onset channels and non-onset channels.
Right.
Yeah, through A1 or phases of A1. Yep.
Right. It's interesting. And does it split into two groups or more than two groups? Are there like unsynchronized state in between?
Unsynchronized state. So so as we can see like briefly in the movie actually, so before the onset of the seizure, you get this essentially like one group of channels, which are the onset channels that are highly desynchronized, but the rest of the channels seem to be pretty synchronized, right? So we kind of group the activities into two groups, right? One synchronized and one desynchronized, and we call that chimera states. But as a seizure happens, for certain seizures, you see a variety of like chaotic behavior. You can see like the flip, in this the flip of this behavior, you could see everything being desynchronized. And so we refer to this as like micro states within the seizure, and we don't yet understand what they represent, but while like consistently before the seizure onset, we seem to see this like clear split between onset and non-onset activities. After the seizure onset, we do see a variety of patterns of synchronization. And that seems to be different across seizures and across patients as well, right? But once the seizure terminates, then we see this like global synchronization of all channels across the brain. So it seems like before the seizure onset and after the seizure termination, we see something more like generalized across seizures and across patients, more or less, right? But with like during the seizure, we see a variety of different combinations of things, and that differ even within the same patient. And we don't yet understand what that means.
One thing I find quite fascinating from the data that you have, I think on page 22, when their onset of this seizure, it seems to be following similar patterns. Are there any good reason why they're split into two groups of neurons and then the seizure start?
So you're asking after the pink arrows, right? You seem to see
right before, right before the arrows. It seems to be the same pattern, split into two groups or whatever, three groups.
Yeah, so it's a little bit right. So that is, that's right. And it what's a little bit tricky about this plot is that we normalize it, right? So we're only plotting like 30% of the channels, and then we normalize it. So then 1 and 0 would show like complete complete synchronization. And so when you see white here, it could be either one, right? It could be, because we want to essentially focus on just the chimera states. Yeah, so that's why I guess it's like a simplified way of quantifying as the partial synchrony, right? And that's why you see these patterns within these regions, but most of the plot is white. Yeah, that's just also the way we like normalize the PDA on the
does the black head of the dragon and the yellow head of the dragons means different things in the brain?
That could be my next project. Did not think about that.
So does it mean that this group of electrodes that having a chimera state is causing the other group to be chaotic after? Like, can you determine that causality?
Yeah, so we cannot really determine causality between channels based on DDA. So as I briefly mentioned, there's an extension of this method that has been developed by Claudia and Chris, and they have tried it on epilepsy data as well. And they also see like, this causality information, like going basically activities going from onset channels to like the rest of the channels. So that adds more like information, regarding kind of the spatial location and the direction of information, right? That gives rise to epileptic seizures within like what regions or what channel, basically. Yeah.
Is this the same as stranger or no?
Sorry?
Is this the same as stranger causality or no?
This?
No, the CDA.
So I'm not an expert on CDDA.
It's not like stranger causality.
Right.
So stranger causality basically has is looking at the AR network, and here we have again X dot goes from one channel and the right side of the equation from the other channel and the other way around, and then you can see the information flow. We also published a paper about this. I
see.
Sorry.
But you might be able to do some causality manipulation here, right? Because you can also introduce electrical stimulation on the electrodes.
Yeah.
So maybe it's possible that if you disrupt the chaotic or synchronous whatever, and see what would happen to the other population.
Yeah. So actually, instead of actually doing that, we can try to induce that in our network, right? So we have, say, 77 nodes, right? You want to do something, you want to, stimulate one node, right? So then you can kind of think about what that would mean to the neighboring channels, for example. And then you can essentially like change the connectivity matrix based on your assumption of what stimulation would do. And then you can look at the network dynamics after you change that connectivity matrix, right, after the stimulation, and compare to what it would look like without the stimulation. And then you can quantify the changes, right, the transition time or level of synchronous ability or something like that. You can just simulate that. And then that should give you at least some information, right, about what like stimulation would cause or what causality.
Right.
Yeah. I think. Yeah.
I think from the data, the onset of CHIMERA states, or the onset of chaos more precisely, seems to follow the same trend. I'm not sure if there's a known theoretical justification behind that. You can show the slides earlier. I don't remember which one. Before this?
You mean?
Before this.
Let's see.
Yeah, this one. Like if you look at the order parameters, they sort of follows the same trend until it gets into this arrow region, right? Seizure, when seizure happens, or whatsoever. I'm just curious, is there any known statistics that we can predict the onset of seizure? Like how close you are to seizure state? Because it looks, I don't know, to my eyes at least, kind of similar. I mean, transition from high DDA to low, and then it starts to kick back into high DDA again at the end of the road.
Right. Yeah, so let me just show this one. So this is the same similar plot, right? But with a different method, like Kuramoto order parameter, just to look at like, you know, the zoom in version, so then it's easier to see. As basically it follows what you were saying before, where you see this like, you know, lowering of like order parameter, right? That goes from like really high value, like highly synchronized, and then so like, and this the black line here indicates the seizure onset as indicated by neurologists, right? But if we were to just use our data, we would say that the onset is like around here, right? And so yeah, but this is also in one patient, right? Like so within this one patient, we could use like, you know, for example, DDA order parameter, like the kind of the scale, like how low the value is, or the flip between lowest value to highest value to predict precisely like when the onset should be, right? But then yeah, like that also changes depending on like what types of seizures, or you know, across patients as well. But yes, at least in this one patient, right? You could say that, like you could say that by tracking like the values of order parameter, you could predict when the seizure should happen.
Right, right. The reason I'm asking is because in statistical physics community, there's a concept of universality. I mean, it can be different patient, the microscopic details of the models can be different, but the approach to phase transitions, which is exactly, I think, what you're looking at right now, where the order parameter goes down and shifts back to almost one. That's the onset of phase transitions. It seems to be universal for many models that we know. I don't know about this CHIMERA models, but yeah, maybe I should take a look, because the universality, it's one nice thing about many body statistical physics. But I think, I'm not sure because you haven't tried with many patients, but I would suspect for different patients, the slope of order parameters change as a function of time. Could potentially be similar, I would guess. I don't know.
I see. Thank you.
Okay. So far, we have only questions for the first part of the talk, right? But that's a long question session already.
Siraj asked a lot of questions about the model, about the network.
All right.
Asking a lot of questions. It's really good. It's really good. I'm so impressed.
Yeah, the data and the analysis are not nice.
Thank you.
Any more questions from the audience? Okay. If no more question, then maybe we should thank Nuttida, Dr. Nuttida again นะครับ ขอบคุณมากครับ That's the only Thai word I would say.
ขอบคุณครับ
Thank you.
Thank you, everyone.
Thank you, everyone.
Thank you.
So I hope this come out as like more collaboration collaboration in the future. And I think we have a lot of resource here, and we have like the team here, like CHIMERA, he is like a mathematical physics, and he's building the team here. Chaipat is building the team there, and I'm building the team here. And in the future, if we have funding or something like that, you guys can come to Thailand and check out our hospitals. And you know, Thailand is hot like the summer, like all year. And right now the hotel is cheap, you know, and
we don't have COVID yet.
I'm totally down.
Yeah, everyone is down. So. May not come tomorrow to California, but we can do something really good.
Thank you. Okay.
All right.
All right. So
All right,
thank you, everyone.
All right, take care, everyone.
I will see you later.
Thank you so much again. We should treat you a meal once you come back to Bangkok.
Yes.
A few meals, because it's hosted by 3 dinners. 3 Thai foods.
Yes, please. Okay.
โอเค
ขอบคุณค่ะ
บ๊ายบาย
ไว้คุยกัน
ขอบคุณ
ค่ะ
มากครับขอบคุณมากครับ สวัสดีครับ
สวัสดีค่ะ